as I said this morning, Dr. Bhaba's vision about how fundamental research and exploratory research in all areas of science on one side can be linked to mission-driven goals is something which is truly inspiring, and we must read a lot of that. And organizations internationally which have succeeded in mission-driven programs have had this kind of interface, and we need to do that in a rapidly changing world. As Dr. Kakodkar mentioned, I'll tell you about, my talk is going to be about life sciences and what it's about and what the future holds, but a lot of it about how it's linked to life sciences and the DAE ecosystem towards the end. But life sciences in the DAE ecosystem started with, as Dr. Kakodkar mentioned, Gopal Iyengar, and he set up a lot of the biology in BARC, what later became BARC, the biology unit, the radiation biology, and the agricultural group also came up under his broad umbrella. And he was quite a remarkable man, and I would urge you all to read about this. About the same time, well, a little later, Leo Szilard wrote to Homi Bhabha suggesting that TIFR start a biology group, and suggested that he look at Ubaid Siddiqui. And Ubaid Siddiqui is here, and Bhabha asked the TIFR faculty what they thought of hiring him, and they were absolutely unanimous that he should not be hired, and that he should be sent to BARC because biology smells. I mean, that was one implication. And fortunately, Bhabha told them that he had already made Ubaid an offer, and therefore he couldn't go back on that. And Ubaid Siddiqui set up the molecular biology unit at TIFR. I mean, this really saved two organizations. It's transformed TIFR into becoming a major center for molecular biology, and later on the growth of NCBS. But it also rescued BARC, because if Ubaid had gone to BARC, it would have been a disaster. He is not a mission mode person. He was not a mission mode person. So this wisdom of keeping people in their places where their talents can blossom is something very important. You put as Dr. Kakodkar's last cartoon showed, you put the right person in the wrong place, then you have a serious problem. The monkey always wins. So my talk is going to be about in praise of useless research and also of useful research. I mean, both of them go hand in hand. Quite often when people talk about being allowed to do useless research, exploratory research, they say, well, this takes a long time to pay off, and unless you invest now, this is not going to pay off, so please do that. Now, there's a problem there when you discuss this with people in Delhi. They will tell you, very good, this is very true, but there are hundreds of years of research happening in the West. Why don't we, at least for now, take the fruits of that and just focus on applied work, and then worry about basic research, which is very expensive, open-ended, no particular goals later on. So we need to have an argument about why India should also focus on fundamental research. And I think TIFR and DAE together overall have given that answer to that, but anyway, I won't have time to go into that in detail. So, what is life, and what should research in life sciences be? Now, this was addressed in a book by Erwin Schrödinger, taking refuge in Dublin from the Second World War, and his book was focused entirely on information in in DNA, and how that information brings translation into the bodies which we see, plants, animals, and so on and so forth. There was another scientist, much later, very recent, still alive and healthy and well, who asked the same question in his book. And that book is also called What is Life? But he didn't restrict himself, Paul Nurse, to looking at just information, but he looked at other aspects which taken together constitute life sciences, and that's very important. And that's very important as in a world today where life sciences and biotech along with artificial intelligence, machine learning, computer science, and with materials research, are going to be truly transformative in the world going ahead. So what is life then? And according to Paul Nurse, life is about cells, about evolution, about DNA, about chemistry, and information. So these are all the multiple components of life, and it's not just DNA. And so how does one integrate all of this to look at life sciences going forward? So let me run through that briefly. So cells. Many people have thought that DNA is the right level of abstraction to understand life sciences and looked upon DNA as encoding life. And indeed, Schrödinger put it in the following way. He says that DNA, he didn't call it that, but chromosomes, he said, are both builder's craft and architect's plan. So the entire plan for creating an organism and the way to construct it are in the DNA. And that's proven multiple times to be incorrect. The right level of abstraction to understand life are cells. Cells are the factory where information from DNA flows in, information from outside comes in, cells divide to form new cells, and understanding cells of all kinds in human bodies, in plants, in the deep ocean, and so on is critically important. Genes are also very important because genes are the thread which links all life on Earth right from the beginning to now by a shared chemistry which I'll come to. And that thread of DNA is what reflects evolutionary history, again which I'll come to. But DNA provides the mechanism by which regulatory influences on cells becoming different types respond to environmental cues and change. So DNA is critically important. And here's a picture of, on the left, a fruit fly with its normal pair of wings, and a mutation in one gene can make this a fly with 4 wings. And this is an example of completely useless research, which over a very short period has completely transformed our understanding of medicine. All of what Tata Memorial Center does in cancer, what we understand about biology, come from this fundamental understanding of how genes and cells work together to make normal animals, and how when mutated or under conditions of stress and strain to cells, they can cause cancer and other illnesses. So the way organisms develop is by signaling to cells to make new kinds of cells, and the principles which are used in a worm or a fly shown on the left side here are pretty much the same principles used in humans. So these so-called model organisms have been invaluable in understanding how humans are made. And today, stem cells, which are again being used in many of our laboratories, our DAE laboratories, are invaluable in actually looking at humans as a model organism, combining our success from molecular and cellular biology from diverse places into understanding humans directly. So this is a very exciting area going ahead. And on the right are shown human genetic diseases which stem from mutations in the kinds of genes which I told you, which make 4-winged flies. So I've talked about chemistry and cells, genes and cells, about chemistry. In cells, you have an extraordinary set of chemistry going on as the consequence of genes which are involved in making proteins, and proteins interacting with other proteins to make metabolites, signaling to other cells and receiving signals, and all this chemistry fundamentally is similar in many, many ways, from the baker's yeast which you use to make beer and bread and so on and so forth, to human. And this has been completely transformative in our understanding of how chemistry works across different organisms, plants, animals, and so on, because you can take chemistry through multiple techniques, put it from one cell to another and understand this. So understanding chemistry, metabolism, how energy supply affects our development and function, all of that has been completely transformative, because how chemistry functions in cells has been very useful. Ultimately, the key aspect of all of this, cells, tissue, chemistry, evolution of the evolution of humans and their chemistry and their genes, all of that moves into information. The reason why organisms function the way they do is because they're enclosed, and therefore they can maintain low entropy against the wall outside, which demands increasing entropy. This requires energy, but this ability of a bacterial cell, for example, to sense the environment and have order inside and respond is something, information flow, which is vital to all of biology. It's true of the bacterial cell, and it's also true of the 86 billion cells which make up our brain. And our understanding of all this information processing is going to be invaluable for every single possible application, and this is a very key area of biology which we need to get into in a substantially greater manner. Now, I told you about one cell. Here are about 50,000 cells which are there in the fly's brain, and each nerve cell and how it's connected to every other nerve cell has been deciphered by a group, a bottom-up group combined with a top-down group working on electron microscopy data and ground truthing of 300 people, including several college students, to understand the entire connectome of the fly. The connectome, how every nerve cell is connected to every other nerve cell, is a link between the sensory world outside and the cognition which goes into the brain and action which comes out in the organism. This is feasible today in the fly, in the worm. It will soon be feasible as technology ramps up in the mouse. Today, 1 cubic centimeter of a mouse's brain will take at least 50 or 100 years to get this kind of a detail, and a human brain will take much, much longer. But technology is changing at such a fast pace that I will not be surprised if a human brain we could get this kind of a resolution in 10 to 20 years. Now, as I said, the point about sensing and cognition is output, and that output comes from movement. Movement is the output of the nervous system, and here's a fly playing football in slow motion. Now, because you can understand what every animal does outside, humans, rats, cats, bats, plants, and so on, by detailing what is called the phenotype, and you have an understanding inside of gene expression, circuit connection, metabolism, you have an enormous amount of information which now allows you to connect what is happening in a personalized way in any organism, in a plant or in a human. So personalized delivery of or understanding at the first level, and then delivery of mediations will be increasingly feasible. Now, all of this understanding came from our exploration of various kinds of organisms in the world, and this is a large squid, and its giant axon allowed us to image and understand, not image, to poke needles and look at the physiology and understand how action potential signals from one end of a nerve cell go to the other end of the nerve cell. And this dramatic change allowed us to understand every single nerve cell and how it functions. It was arduous and very, very laborious process, and that's been transformed today by our ability to image hundreds and thousands of nerve cells simultaneously. So when you look at a brain slice, you can see what is happening in terms of physiological activity simultaneously in a very large number of cells. So this again is contributing to the data, and understanding needs to flow from this large-scale data collection. So cells, genes, evolution, chemistry, and information are all very important for all organisms of Earth and how they function, but also, of course, for the human brain. So the human brain needs to do two things. Our understanding of the world outside, our understanding of nature's engineering comes from the capacity of the brain. But our understanding of our own brain and how the brain has been engineered is very, very low. And that is going to be a really big and challenging project. The Tata Memorial Hospital has got large cohorts looking at cancer in Barshi. Chris Gopalakrishnan has started the Center for Brain Research, and they have large cohorts. The Mohan Diabetes Center in Chennai has again large cohorts, as have other cohorts in Delhi. These cohorts follow people over time and look at their various parameters from their own perspective and see how whether cancer or blood pressure or diabetes or stroke comes about. Combining that with a cognitive assay and combining all these cohorts and partnering with each other allows an understanding of brain function, but also impressively, very impressively, a movement from the most fundamental of research to important applications with great speed. Now, we've often looked at fundamental research as something and application as something else. Life, unfortunately, doesn't work that way. Life is a jalebi. There is, a constant feedback. If you look at history, people who have done applied research, the discovery of DNA as a hereditary molecule came from looking at sewage and seeing what is the basis of infection of Streptococcus pneumoniae, right? And the transforming principle turned out to be DNA. DNA from one kind of bacteria could be put into another and change its properties. That came from completely applied search. And many, many fundamental discoveries have had extraordinary applications. And many people, many labs, many groups have done both simultaneously. And one big example is the way the nuclear effort, both before, during, and after the Second World War took place all over the world. That's been again very transformative. So we need to see a mechanism by which this is seamlessly allowed and encouraged in DAE institutions. Right now it is encouraged on paper, but in practice, for a person at a DAE institution to start a company, compared to the effort it takes in a Ministry of Education ministry, the uncertainties and the hills they have to cross are far too high. We have to learn from our Ministry of Education colleagues and see how we can encourage that on scale. Make it rewarding, encouraging to do that, not by having ticks and boxes ticked about what you need to do, but making this seamless connect very valuable. And that's something very feasible. How do we go about our work now, Alain, in the DAE family? At the bottom is what I would call, research which is completely curiosity driven. And I must stress before I go on to the other points that these different layers are connected, with each other. That should be encouraged widely and should have wide connection with core DAE missions. And I alluded this morning about ways of doing that. I will not get into that in greater detail, but this needs to be encouraged enormously. While that is happening in many of our autonomous institutions, we must also keep in mind, even in India, the numbers of autonomous institutions of various other organizations have grown by leaps and bounds. They are populated by students, former students and former postdocs of all of you, and they are now doing what you are doing, and therefore you need to stand out substantially. You can't do more of the same. If you do more of the same, even if you do it efficiently, you will get more of the same. The world is changing very rapidly, and this needs to be, again, extremely aggressively combative. Just because it's open-ended doesn't mean it's not goal-driven. And there, collaborations in a wise way, as Dr. Kakotkar says, international links to our institutions, openness to handle that, yet a sense of one's own purpose is needed. And there are very fantastic examples in our ecosystem about how that has been done. You'll hear Ashok Sen, for example, Suman Mahan. Now, that kind of a purposeless work, as it were, and this is an example of a fly from Sanjay Sane's lab. On one side, on the left flapping its wing, on the right flapping what's called a halter, a structure for balancing with high resolution camera. And our own group is studying how the insides of that actually develop. Now, this is ostensibly completely purposeless science, but it involves collaboration both within our laboratories, between other laboratories, international laboratories, and with people working on robotics which are bio-inspired. And so this has taken a speed of its own which was unimaginable even 10 years ago. So the pace of application of completely useless research to usefulness is changing dramatically, and each of us can grasp it if we want to. The second layer of research is various kinds of collaborations which are slightly better organized bottom up and top down, and there are very good examples of that in the DAE system. One is the famous Samba Masuri rice. This has transformed Indian rice agriculture enormously. And one measure of its success is that if you go to the Department of Biotechnology, they'll say that they were responsible for Samba Masuri. You go to CSR, they say they're responsible for Samba Masuri. You go to ICR, they say they're responsible for Samba Masuri. And DAE has played a great role. So success has many fathers. And this is an example of how successful work can be done by collaboration between different agencies. And DAE really has driven, DAE's, BRC's agriculture division has driven this in an extraordinarily positive and very valuable way. And we should all be very proud of that. So this is an example of, not bottom up, not top down, but a mid-level collaboration. At the top down there are missions, and those missions need to take on big projects. It's, as the Chinese general said, if something will take 20 years to get done, the right time to start is now. And these big projects involve understanding the brain and its function. This brain takes up 20 watts of power and can do extraordinary things. Understanding how it functions in health and disease, how we can deal with an aging population and look at mobility issues, dementia and so on is a very exciting project. And there are many, many other exciting projects in agriculture. Agriculture of the next generation will be enormously technology driven, where soil, fertilizer, pesticides are minimally used, where climate regulation are controlled enormously, yield is high despite and pest interference is low. So these are enormous demands which require the capability of the BARC system, which has got expertise, and the IG card system has got expertise in other areas, but will be related enormously to this. So this, in terms of methane emissions, the kinds of yield, is a very, very important area, and coordination between fundamental and applied research in agriculture is another area. Bioenergy is another one. There are multiple sources of energies in our portfolio which we need, and bio-waste handling is going to be a big time affair. DAE has already put in an enormous amount of effort in this, as have other agencies, and interministerial collaboration here. I wouldn't say, I don't know whether I should say led by DAE or not, but if DAE is a player at the high table, we can be sure that this will be effective. Our understanding of cells in our environment, whether it is in our backyard or anywhere else, is enormously poor. And the tools for this understanding, genomic, mass spectrometry, expansion microscopy, are just enormous. There is enormous basic research to be done here, and enormous intellectual property to be got. And we have the capability distributed in our laboratories to do this. We are not working together in a focused manner on big missions in these kinds of areas. Astronomers, a dozen of them, will get together and make a strong and compelling case for a 30-meter telescope. Biologists, 500 of them, will not make a compelling case for a coordinated mission of importance. Unless biologists learn to behave like astronomers, they are going to be subsumed into being second-rate players in a first-rate world. So this is something very important for life scientists to consider. You want a tuberculosis understanding or a malaria understanding, these have to be done in a mission mode and not in a distributed manner. And similarly with microbiology of various kinds. The deep ocean, I'll end with this, my last slide, is an exciting and enormously promising area which the Ministry of Earth Sciences is leading the deep ocean mission. The DAE is a partner. BARC is working on some of their verticals. This is going to be as valuable, I would say, maybe more, because our understanding of the deep ocean is actually much less than our understanding of space, of outer space. The first slide I show were tube worms which are resident in deep ocean hydrothermal vents. They have no mouth and they have no butt, and they have resident bacteria inside them which take chemicals from deep ocean vents and process them to give energy to the world. These kinds of new biology is there everywhere, new organisms are there to be discovered, new ways of functioning, new minerals. It's an enormous opportunity which is so important that it shouldn't be left to the Ministry of Earth Sciences alone. All of us should also partner. So thank you very, very much for this. I'd like to end for one quick point that again, we must keep in mind Homi Bhabha's vision of combining, as I said this morning, of what Edward Teller and Bhabha said about having open interaction of our closed structures with the open system, free for all from all over the world, and linking that with the internal work so that big missions inside can stand. And this is a image of a synapse of getting inputs, of a neuron getting inputs from trillions of, tens of thousands of inputs coming in, and the output then for our missions would be very valuable. Thank you very much.
Okay, sir. So we should avoid doing more of the same, even that is